Hey guys, welcome to this news story. Today, we look at something that happens to every single person on this planet once a month, whether they notice it or not. The full moon rises, bright and round and hard to ignore, and something inside the human brain shifts with it. People have said this for thousands of years, across cultures, across continents, across centuries of recorded history. Sailors, doctors, farmers, and philosophers all noted the same pattern long before modern science had the tools to test it. For most of that time, the idea was dismissed as folklore, a superstition, the kind of thing that sounded meaningful but could not hold up under real scrutiny. Then the studies started coming in. Sleep data collected over years showed people falling asleep later and sleeping less around the full moon, even in rooms with no windows. Mood tracking research pointed to shifts in anxiety and restlessness that followed the lunar cycle with uncomfortable consistency. Brain activity, hormone levels, and behavioral patterns in study after study kept showing small but real changes timed to the moon's phases. So, what is actually happening? How does a rock 250,000 miles away reach into the human body and move something? What biological mechanism could possibly connect the moon's cycle to the rhythms running inside us? In this video, we go through the research, break down what the data actually shows, and ask the question that cuts right to the heart of it. How much of what we feel every month has always been written in the sky above us? The moon is not a small object. It measures about 3,474 kilometers across. That is roughly the distance from the western edge of the United States to its eastern coast. When people look up at the night sky and see the moon hanging there, it is easy to think of it as something light and distant, almost decorative. But the moon is a solid, dense, rocky body with a mass of about 7.35 times 10 to the power of 22 kilograms. That number is difficult to picture in practical terms. To put it another way, the moon contains enough mass that its gravity reaches across nearly 385,000 kilometers of space and pulls on every ocean, every lake, and every drop of liquid on Earth with measurable force. The moon sits, on average, about 384,400 kilometers away from Earth. That distance is not fixed. The moon travels in an elliptical path around Earth, meaning the distance changes slightly throughout each orbit. At its closest point, called perigee, the moon comes to about 356,500 kilometers away. Its farthest point, called apogee, that distance stretches to around 406,700 kilometers. This variation matters more than most people realize. When the moon is closer, its gravitational pull is stronger. When it is farther away, that pull weakens. The difference is not dramatic enough to cause catastrophic changes on Earth, but it is large enough to be measured and large enough to influence the biological systems of living things. To understand how the moon came to be where it is, it helps to go back about 4.5 billion years. The solar system was forming from a large spinning disk of gas and dust. Earth was still young, still largely molten, and still being shaped by constant collisions with other rocky bodies drifting through the early solar system. One of those bodies was roughly the size of Mars. Scientists call it Theia. Around 4.5 billion years ago, Theia collided with the young Earth at an angle. The collision was not head-on. It was more of a glancing blow, and the energy released was enormous. A large portion of Earth's outer layers was blasted away into space. Much of Theia itself was destroyed in the impact. The material thrown into orbit around Earth, a mix of vaporized rock and molten debris, gradually clumped together under its own gravity. Over millions of years, this debris cooled, solidified, and formed the moon. This origin story, known as the giant impact hypothesis, is supported by several lines of evidence. The moon's composition closely matches Earth's outer layers. The ratio of oxygen isotopes in moon rocks brought back by Apollo missions is nearly identical to those found in Earth's mantle. The moon has a relatively small iron core compared to its total size, which makes sense if it formed mostly from the rocky outer layers of Earth rather than from its iron rich interior. No other explanation for the moon's formation fits the evidence as well as this one does. After the moon formed, it was much closer to Earth than it is today. Scientists estimate that early on, the moon may have been as close as 20,000 to 30,000 kilometers away. At that distance, its gravitational effect on Earth would have been far more powerful than anything experienced today. The tides it created in Earth's early oceans would have been enormous. The moon has been gradually moving away from Earth ever since, at a rate of about 3.8 centimeters per year. That rate is tiny, but over billions of years it adds up. The moon continues to move away slowly, and this slow retreat is directly connected to the way the moon and Earth exchange rotational energy through tidal forces. The surface of the moon tells a long and detailed story. It is covered in a layer of broken rock and fine dust called regolith. This layer formed over billions of years as meteorites, asteroids, and smaller particles struck the surface, grinding the rock into smaller and smaller pieces. The regolith can be anywhere from a couple of meters deep in some places to tens of meters deep in others. Below the regolith lies a thick layer of fractured rock called the megaregolith, which itself sits on top of the ancient lunar crust. The moon's surface is divided into two broad types of terrain. The first is the bright, heavily cratered highlands. These are the oldest parts of the moon's surface, dating back more than 4 billion years. The second type is the dark, smoother regions called Maria, which is the Latin word for seas. Early astronomers thought these dark patches might actually be bodies of water, which is how they got the name. They are not water. They are ancient lava plains, formed between about 3 and 3.5 billion years ago, when the moon was still geologically active enough to produce volcanic eruptions. Lava flowed out from the interior, flooded large low-lying basins, and cooled into flat dark plains of basaltic rock. The largest of these Maria, the Oceanus Procellarum, or Ocean of Storms, stretches across roughly 2.5 million square kilometers. On the moon's near side, that is larger than the Mediterranean Sea. Other large Maria include Mare Imbrium, Mare Serenitatis, and Mare Tranquillitatis, which is the Sea of Tranquility, the site where the Apollo 11 mission landed in 1969. These names, given by early astronomers, have stuck even though the regions contain no water at all. The moon's craters are among the most immediately visible features of its surface. They range in size from tiny pits a few millimeters across to enormous impact basins hundreds of kilometers in diameter. The South Pole Aitken Basin, one of the largest and oldest impact craters in the solar system, is about 2,500 kilometers wide and more than 8 kilometers deep. It sits on the moon's far side, largely hidden from Earth. Most of the moon's craters formed during a period called the Late Heavy Bombardment, roughly 4.1 to 3.8 billion years ago, when the inner solar system was being pelted by a large number of asteroids and comets. Earth went through the same bombardment, but Earth's geological activity, erosion, and plate tectonics have erased most of that evidence. On the moon, with no weather and no plate movement, those ancient craters remain almost perfectly preserved. The moon has no atmosphere in the way Earth does. There's an extremely thin layer of gases at the surface, sometimes called an exosphere, made up of small amounts of hydrogen, helium, sodium, and potassium. But this layer is so thin that its molecules rarely interact with each other. It provides no weather, no wind, no protection from solar radiation, and no insulation against temperature changes. As a result, the moon's surface experiences extreme temperature swings. During the lunar day, the surface in direct sunlight can reach about 127 degrees Celsius. During the lunar night, the temperature drops to around negative 173 degrees Celsius. These are not gradual transitions, they happen over the course of a lunar day, which lasts about 29.5 Earth days. Because The moon has no atmosphere to scatter or absorb light. The sky on the moon is always black, even when the sun is up. There is no blue sky, no twilight, no diffuse glow. Shadows are sharp and perfectly dark. The contrast between sunlit surfaces and shadows is extreme. This characteristic of the moon's environment has practical consequences for anyone or anything on its surface, and it also explains certain features of how moonlight behaves when it reaches Earth. The moon rotates on its axis at almost exactly the same rate as it orbits Earth. This is not a coincidence. It is the result of tidal locking, a process in which the gravitational interaction between two bodies slows the rotation of the smaller one until it matches its orbital period. Over millions of years, Earth's gravity gradually slowed the moon's rotation until the moon's day and its orbit around Earth were the same length. The result is that the same side of the moon always faces Earth, the near side, which is what every person who has ever looked up at the moon has seen, is the only side visible from Earth's surface. The far side, often called the dark side, though that name is misleading because it receives just as much sunlight as the near side, remained completely unseen by human eyes until 1959, when the Soviet spacecraft Luna 3 flew around the moon and sent back the first photographs. The moon's interior has been studied using seismometers placed on the surface during the Apollo missions. Those instruments detected moonquakes and allowed scientists to map the moon's internal structure. The outermost layer is the crust, which varies in thickness from about 50 kilometers on the near side to about 60 kilometers on the far side. Below the crust is the mantle, a thick layer of rock that extends deep into the interior. At the very center is a small core. The inner core is solid and made mostly of iron, measuring about 240 kilometers in radius. Surrounding it is a fluid outer core, also iron rich, extending to about 330 kilometers in radius. This small, partially molten core means the moon generates a very weak magnetic field, far weaker than Earth's. In the distant past, the moon may have had a stronger magnetic field when its core was more active, but that activity has largely ceased. The moon's gravity, measured at its surface, is about 1.62 meters per second squared. Earth's surface gravity is about 9.81 meters per second squared. This means the moon's surface gravity is roughly 1/6 that of Earth. An object that weighs 60 kilograms on Earth would weigh about 10 kilograms on the moon. This lower gravity is directly tied to the moon's smaller mass. It also means the moon cannot hold onto a thick atmosphere because gas molecules move fast enough at the surface temperatures to escape the moon's weaker gravitational pull. Water ice has been confirmed to exist on the moon, particularly in permanently shadowed craters near the poles. These craters are so deep and their walls so steep that sunlight never reaches their floors. In that permanent darkness, temperatures can drop to around negative 250 degrees Celsius, cold enough to trap and preserve water ice for billions of years. The Lunar Reconnaissance Orbiter, operating since 2009, has mapped these cold traps in detail, and spectral data has confirmed the presence of water ice. The total amount of water ice locked in these polar regions is not yet precisely known, but estimates suggest it could be in the range of hundreds of millions of tons. This water ice is significant not only scientifically, but also practically, as future lunar missions are likely to consider it a resource. The moon's orbital mechanics are worth examining in some detail because they directly determine what happens in the sky above Earth. The moon orbits Earth in a slightly tilted plane, about 5.1 degrees relative to the plane of Earth's orbit around the Sun. This tilt is why eclipses do not happen every month. If the moon's orbit were perfectly aligned with Earth's orbital plane, there would be a solar eclipse every new moon and a lunar eclipse every full moon. Because of the tilt, the moon usually passes above or below the alignment needed to create an eclipse. Eclipses only occur when the moon's orbit intersects Earth's orbital plane at just the right moment during a new or full moon. the moon's orbit is also slowly shifting over very long timescales. The gravitational influence of the sun and other planets causes the orientation of the moon's orbit to change gradually over cycles of thousands of years. These long cycles affect the frequency and character of eclipses and have been tracked and calculated by astronomers for centuries. One of the more striking facts about the moon's current position in the solar system is related to a remarkable coincidence of size and distance. The sun is about 400 times larger in diameter than the moon. The sun is also about 400 times farther from Earth than the moon. These two numbers being nearly equal means that from Earth's surface, the sun and the moon appear almost exactly the same size in the sky. This is why total solar eclipses are possible at all. When the moon passes directly between Earth and the sun during a new moon, it can block the sun's disk almost exactly, creating the dramatic ring of the solar corona visible during totality. This apparent size match is a coincidence of the current moment in Earth's history. Because the moon is slowly moving away, in the distant future the moon will appear smaller in the sky and will no longer be able to fully cover the sun. The moon also plays a direct role in stabilizing Earth's axial tilt. Earth's axis is currently tilted at about 23.4 degrees relative to its orbital plane. This tilt is what creates the seasons. Without the moon's gravitational stabilizing influence, simulations suggest Earth's axial tilt could vary much more wildly over millions of years, potentially swinging between near zero and much larger angles. Such large variations in tilt would produce dramatic climate shifts, potentially making Earth's climate far less stable over geological time. The moon, by exerting a steady gravitational torque on Earth's equatorial bulge, helps keep the axial tilt within a relatively narrow range. This stabilizing role is one reason some scientists consider the moon to be an important factor in the long-term habitability of Earth. The moon reflects sunlight back to Earth with relatively low efficiency. Its surface is dark on average, reflecting only about 12% of the sunlight that strikes it. This reflective property is called albedo. An albedo of 12% is actually quite low. Fresh snow reflects about 80 to 90% of sunlight. The moon's dark volcanic basalt and ancient dust absorb most of the light that hits them. Despite this low reflectivity, the full moon is bright enough on Earth's surface to cast shadows at night, to make color vision possible. In some cases, and to produce enough illumination for animals, including humans, to navigate and carry out activity without any other light source. The full moon produces an illuminance on Earth's surface of about 0.1 to 0.3 lux. For comparison, a typical indoor office is lit to around 300 to 500 lux. A clear midday in direct sunlight produces about 100,000 lux. Moonlight is far dimmer than any of these, but it is not negligible. Many biological systems have evolved to detect and respond to illumination levels far lower than what the full moon provides. The fact that moonlight is bright enough to matter biologically, while still being orders of magnitude dimmer than daylight, puts it in a particular zone of the light environment that living systems have had to contend with throughout evolutionary history. The moon orbits Earth roughly every 27.3 days relative to the stars. This is called the sidereal month. But because Earth is also orbiting the sun during that time, the moon needs slightly longer, about 29.5 days, to return to the same phase as seen from Earth. This is the synodic month, the period most people know as the lunar month or the time from one full moon to the next. The difference between 27.3 and 29.5 days comes from the fact that Earth moves about 27 degrees along its orbit around the sun in that time, so the moon has to travel that extra distance to realign with the sun as seen from Earth. This 29.5-day cycle is one of the most consistent and observable natural rhythms available to living things on Earth. It has been present and largely unchanged for billions of years. It repeats without interruption. It is tied to measurable changes in light, in gravitational force, and in the geometry of the Earth Moon Sun system. Many organisms, from microscopic plankton to large mammals, have internal timing systems that run on roughly monthly cycles, and researchers continue to investigate how much of this biological monthly rhythm is tied to the lunar cycle versus other internal or environmental factors. The lunar cycle begins at a point of near invisibility. When the moon sits between Earth and the sun, its sunlit side faces completely away from Earth. From the ground, the moon is present in the sky, but almost no reflected light reaches the surface. This is the new moon. The moon is not gone. It is simply dark from Earth's perspective, moving through the sky during daylight hours, largely undetectable without specialized equipment. From this point, the cycle moves forward day by day. As the moon continues along its orbit, it shifts eastward relative to the sun in the sky. A thin sliver of its sunlit side becomes visible from Earth after about 1 to 2 days. This crescent is narrow at first, just a slim curve of reflected light low in the western sky after sunset. Each day that passes, the moon moves farther along its orbit, and a slightly larger portion of the sunlit face turns toward Earth. The crescent grows. By about 7 days into the cycle, the moon has traveled roughly 1/4 of the way around Earth. At this point, exactly half of the moon's visible face is lit by the sun. This is the first quarter moon. The name refers to the orbital position, not the amount of light visible. Half the face is lit, but the moon is 1/4 of the way through its cycle. This phase is visible in the sky during the afternoon and the evening, setting around midnight. After the first quarter, the lit portion continues to expand. The moon is now described as waxing, meaning growing. The lit area moves past the halfway point, and the shape becomes what is called gibbous, a term that simply means more than half lit, but not yet fully round. The waxing gibbous moon rises in the afternoon, climbs high during the evening hours, and sets in the early morning. It is bright enough to cast light across open ground and reduce the visibility of dimmer stars in its vicinity. Around day 14 to 15 of the cycle, the geometry of the Earth, Moon, and Sun reaches a specific alignment. The Moon is now on the opposite side of Earth from the Sun. The Sun's light falls on the Moon's near side, the side that always faces Earth, almost directly head on. The result is a fully illuminated disk. This is the full moon. The entire near side of the moon is lit at once, and from Earth the moon appears as a complete circle of reflected light rising in the east around the time the sun sets in the west. The full moon's brightness comes entirely from reflected sunlight. The moon produces no light of its own. What reaches Earth is solar radiation that has traveled about 150 million kilometers from the sun, struck the moon's surface, and bounced back across roughly 384,000 kilometers of space to reach the ground. This reflected light travels at the speed of light, about 300,000 kilometers per second, so it takes approximately 1.3 seconds to travel from the moon to Earth. The sunlight that illuminates the full moon left the sun about 8 minutes earlier. The color of moonlight is actually very close to white, with a very slight warm tint caused by the composition of the moon's surface minerals. The lunar regolith, that layer of fine broken rock and dust, contains minerals such as feldspar, pyroxene, and olivine. These absorb certain wavelengths of light more than others, giving the reflected light a marginally warmer tone than pure white. When the full moon sits low on the horizon, it can appear orange or red. This is not a change in the moon itself. It is caused by Earth's atmosphere. At low angles, light from the moon passes through a much thicker slice of atmosphere to reach an observer's eyes. The atmosphere scatters shorter blue wavelengths away and allows the longer red and orange wavelengths to pass through more easily. The same effect makes sunsets appear red and orange. The full moon appears larger when it sits near the horizon than when it is high in the sky. This is a perceptual effect, not a physical one. The moon's actual angular size in the skies does not change significantly as it rises. What changes is the brain's interpretation of size relative to nearby objects. When the moon is near the horizon, it is seen alongside trees, buildings, and terrain features that provide a sense of scale. High in the sky, there are no reference points, and the brain tends to perceive it as smaller. Measurements confirm that the moon's angular diameter at the horizon and at its highest point are nearly identical. The horizon moon illusion has been studied for decades and remains one of the more well documented examples of size perception being influenced by context. After the full moon, the cycle continues. The lit portion begins to shrink. Now the moon is waning. The shape passes back through gibbous, then reaches the third quarter phase around day 22, when half the face is again lit, but now the opposite half from the first quarter. The crescent returns, this time on the left side in the northern hemisphere, narrowing each day as the moon approaches new moon again. By day 29 or 30, the cycle completes, and the moon is once again sitting between Earth and the sun, its near side dark, the cycle ready to begin again. The monthly rhythm of light this creates is not subtle. The difference in nighttime illumination between a new moon night and a full moon night is dramatic. On a clear night with a full moon, the ground level light is sufficient for many animals to see clearly, to hunt, to navigate, and to detect movement at distances that would be impossible on a dark night. The monthly swing from darkness to brightness and back again is one of the most reliable environmental rhythms on Earth, and it has been present, largely unchanged in its basic structure, for hundreds of millions of years. Gravity does not stop at a fixed boundary. It extends outward from every object that has mass, growing weaker with distance but never fully disappearing. The moon, despite being nearly 385,000 kilometers away, exerts a gravitational pull on Earth that is measurable, consistent, and responsible for some of the most visible, large-scale changes on the planet's surface. The basic principle behind tidal force is not simply that the moon pulls on Earth. It is that the moon pulls on different parts of Earth with slightly different strengths. The side of Earth closest to the moon is pulled more strongly than the center of Earth, and the center of Earth is pulled more strongly than the far side. This difference in pull across the diameter of Earth is what creates tides. The water on the side facing the moon is drawn slightly toward it, creating a bulge. On the opposite side of Earth, the water is pulled less strongly than the solid Earth beneath it, so it effectively lags behind, creating a second bulge on the far side. These two bulges of water, one facing the moon and one facing away, are the tidal bulges. As Earth rotates beneath them, different coastlines pass through regions of high water and low water roughly twice a day. The actual rise and fall of tidal water at any given coastline depends on many factors. The shape of the seafloor, the width and depth of bays and inlets, the angle of the coastline relative to the tidal flow, and the local geography all modify how strongly and how quickly tides express themselves. In the open ocean, the tidal rise and fall is relatively small, often less than a meter. But in certain coastal locations, the geometry of the land funnels and amplifies the tidal movement dramatically. The Bay of Fundy on the eastern coast of Canada is the most extreme example on Earth. The bay is long and narrow, and its shape causes tidal resonance, where the natural back and forth movement of water in the bay aligns with the tidal period. The result is a tidal range of up to 16 meters, the largest in the world. At low tide, large areas of seafloor are fully exposed. At high tide, the same areas are under more than a dozen meters of water. The sun also contributes to tidal forces on Earth, though its effect is smaller than the moon's despite the sun being far more massive. Tidal force depends not just on the mass of the object doing the pulling, but also on how sharply its gravitational pull changes across the diameter of Earth. Because the sun is so far away, even though it is massive, the difference in its pull across Earth's relatively small diameter is less pronounced than the moon's difference across that same diameter. The moon's tidal force is about 2.2 times stronger than the sun's. When the sun and moon are aligned during new moon and full moon, their tidal forces combine. The result is called a spring tide, which produces higher high tides and lower low tides than usual. When the sun and moon are at right angles to each other relative to Earth during the quarter moon phases, their forces partially cancel, producing smaller tidal ranges called neap tides. Tidal forces do not only act on water. The solid rock of Earth's crust also deforms under the moon's gravitational pull. This is called the solid Earth tide or the body tide. The ground beneath any given location actually rises and falls by as much as 20 to 30 centimeters twice a day as the tidal bulge passes through the planet. This movement is far too small to feel, and it happens slowly enough that it produces no shaking or vibration. But it is real and measurable. Precision instruments used in geodesy, the science of measuring Earth's shape and dimensions, have to account for this constant tidal deformation in their calculations. Ah, GPS satellites and the ground stations that support them must correct for the body tide to maintain accurate positioning data. The moon's gravitational pull also affects Earth's rotation. As Earth spins and the tidal bulge moves across the surface, friction between the ocean water and the seafloor slows Earth's rotation very slightly. This effect is small on human timescales, but over billions of years it has added up significantly. When the moon first formed, Earth was spinning much faster. A day on early Earth may have been as short as 6 hours. The tidal braking effect has gradually slowed Earth's rotation to the current 24-hour day, and it continues to slow it at a rate of about 1.4 milliseconds per century. This is why scientists must occasionally add a leap second to atomic clocks to keep them aligned with the actual rotational period of Earth. As Earth's rotation slows, angular momentum is transferred to the moon. This is why the moon is gradually moving away from Earth. The energy lost from Earth's rotation does not simply disappear. It goes into the moon's orbit, pushing the moon into a slightly larger, more distant path around Earth. The rate of recession is small, about 3.8 centimeters per year, but it has been measured precisely using laser ranging experiments. Apollo astronauts left retroreflectors on the moon's surface, and scientists on Earth regularly fire laser pulses at these reflectors and measure the time it takes for the light to return. The data confirms the moon is moving away, consistent with the tidal braking model. The oceans are not the only fluid on Earth responding to the moon's gravity. The atmosphere also experiences tidal forces. Atmospheric tides are much smaller in effect than ocean tides, but they are detectable. Air pressure at any location on Earth rises and falls slightly in response to both the moon's gravitational pull and the heating effect of the sun. The lunar atmospheric tide is small enough that it is largely masked by weather patterns, but it can be extracted from long-term pressure records using careful statistical analysis. The cumulative effect of the moon's gravitational reach on Earth is therefore not limited to the visible rise and fall of ocean water at a shoreline. It extends into the solid rock underfoot, into the upper atmosphere, into the speed at which the planet spins, and into the gradual evolution of the Earth-Moon system over geological time. These are not theoretical effects. They are measured continuously, factored into satellite operations, tide prediction models, and the management of atomic time standards. The moon's gravitational presence is woven into the physical operation of Earth at scales ranging from individual coastlines to the planet's long-term rotational history. Light is information. Every photon that reaches the eye carries data about the environment, and the biological systems inside the human body have spent hundreds of millions of years learning how to read that data and respond to it. Moonlight is part of that information stream. It arrives at Earth's surface every night the sky is clear. It changes in intensity on a predictable monthly schedule, and living systems, including the human body, have mechanisms that detect and respond to it in ways that science is only beginning to fully document. To understand how moonlight affects biology, it helps to first understand what moonlight actually is at a physical level. The moon produces no light of its own. What reaches Earth from the moon is sunlight that has traveled about 150 million kilometers from the sun, struck the moon's surface, and reflected back toward Earth. During that reflection, the light changes slightly. The moon's surface is made of dark volcanic rock and fine mineral dust, and these materials absorb some wavelengths of light more than others. The reflected light has a spectral composition that is slightly different from direct sunlight. It is somewhat warmer in tone, meaning it contains a slightly higher proportion of longer red and yellow wavelengths relative to the shorter blue ones. But this difference is subtle. Moonlight is fundamentally reflected sunlight, and its spectral profile is close enough to sunlight that the eye's photoreceptors respond to it in similar ways, just at a vastly lower intensity. The intensity difference between direct sunlight and full moonlight is large. Full moonlight at Earth's surface produces about zero point 0.1 to zero point 0.3 lux of illumination. Direct midday sunlight produces around 100,000 lux. This means full moonlight is roughly 400,000 times dimmer than direct sunlight. Yet this level of light is not nothing. The human eye is remarkably sensitive across a wide range of light levels, and the visual system has two main types of photoreceptor cells in the retina that operate across different parts of this range. Cone cells are concentrated in the central part of the retina in an area called the fovea. They are responsible for color vision and fine detail, and they work well in daylight and in brightly lit environments. There are three types of cones, each sensitive to different ranges of wavelength, roughly corresponding to red, green, and blue light. Cones require a relatively high level of light to function effectively. In dim conditions, cone activity drops off and color discrimination becomes difficult. Rod cells are far more numerous than cones. The human retina contains about 120 million rod cells, compared to about 6 million cones. Rods are distributed across most of the retina, with a higher concentration toward the edges. They are extremely sensitive to low levels of light, capable of detecting just a few photons at a time. Rods do not distinguish color. They respond to light intensity across a broad range of wavelengths, with peak sensitivity around 498 nanometers, which falls in the blue-green part of the spectrum. Under full moonlight, it is primarily the rod cells that are active. The visual image they produce is less sharp than cone-based vision and completely without color, but it is functional. Under a clear full moon on open ground, a person can detect movement, navigate terrain, and identify large objects without any artificial light. The adaptation of rod cells to low light levels takes time. When moving from a brightly lit environment into darkness, the rods gradually increase their sensitivity over a period of about 20 to 30 minutes. This process is called dark adaptation, and it involves the regeneration of a light-sensitive pigment in the rod cells called rhodopsin. Rhodopsin breaks down when exposed to bright light and slowly rebuilds in darkness. Once fully dark adapted, the human eye can detect light levels roughly 100,000 times lower than what it can detect immediately after exposure to bright light. Full moonlight sits well above the threshold of dark adapted rod vision, meaning a person with fully adapted eyes can see reasonably well under a full moon. Beyond the rods and cones, there is a third type of photoreceptor in the human eye that plays a specific and important role in how the body responds to light. These are called intrinsically photosensitive retinal ganglion cells, often shortened to IPRGCs. They were identified and described in detail only in the early 2000s, unlike rods and cones, which are primarily involved. In creating visual images, IPRGCs serve a different function. They detect the overall level of ambient light in the environment and send that information directly to a small structure deep in the brain called the suprachiasmatic nucleus. The suprachiasmatic nucleus, or SCN, sits in the hypothalamus, a part of the brain that manages many of the body's automatic functions. The SCN is essentially the body's central clock. It coordinates the timing of dozens of biological processes, including sleep, hormone release, body temperature, digestion, and cell division, aligning them with the 24-hour cycle of day and night. The SCN does this by receiving light information from the IPRGCs and using it to calibrate the body's internal timing against the actual external light environment. This calibration process is called entrainment. The body's internal clock runs on roughly a 24-hour cycle on its own, but without regular light input, it drifts slightly. Daily light exposure, particularly in the morning and evening, keeps the internal clock synchronized with the actual solar day. The IPRGCs are particularly sensitive to short wavelength blue light with peak sensitivity around 480 nanometers. This is close to the peak sensitivity of rod cells, but distinct from it. The reason this matters is that blue light is the specific signal the SCN uses to determine whether it is daytime or nighttime. High levels of blue light indicate daytime. Falling blue light levels in the evening signal to the SCN that night is approaching, and the SCN responds by triggering the pineal gland, a small gland in the brain, to begin producing a hormone called melatonin. Melatonin is often described simply as a sleep hormone, but that description is somewhat incomplete. It is more accurate to call it a darkness hormone. The pineal gland begins releasing melatonin into the bloodstream when light levels drop in the evening, typically starting 1 to 2 hours before a person's natural sleep time. Melatonin levels peak in the middle of the night, usually between 2 and 4 in the morning, and then fall in the hours before dawn as light begins to increase. Melatonin does not force sleep directly. It signals to the body that it is nighttime, coordinating a range of biological processes that prepare the body for rest. Body temperature drops slightly, blood pressure decreases, the digestive system slows, the immune system shifts some of its activity into repair and maintenance processes. The relationship between light and melatonin is direct and well established. Even relatively low levels of light can suppress melatonin production if they contain enough short wavelength blue content. Studies have shown that light levels as low as 30 to 50 lux, well below normal indoor lighting, can partially suppress melatonin if a person is exposed to it in the hours before sleep. This is the basis for concerns about screen use before bedtime, since phones, tablets, and computer screens emit significant amounts of blue light. Full moonlight at zero point 0.1 to zero point 0.3 lux falls well below the threshold that most laboratory studies have identified as necessary to suppress melatonin significantly. In controlled settings, direct exposure to moonlight alone does not appear to reduce melatonin to the same degree as electric light. However, the situation is more complex in real-world conditions. Moonlight reflected off pale surfaces such as sand, snow, or light-colored stone can be amplified. A person sleeping in an environment with large reflective surfaces, thin curtains, or no curtains at all may be exposed to more total light energy from a full moon than a simple open-air measurement of moonlight intensity would suggest. There is also the question of cumulative sensitivity. Laboratory thresholds for melatonin suppression are typically measured in people who have been exposed to normal indoor light levels throughout the day and evening, in people who live or spend significant time outdoors in natural light conditions with lower overall artificial light exposure. The visual system may be more sensitive to lower light levels, including moonlight. Research on this specific question is limited, but it is a recognized variable in studies examining how natural light environments affect sleep and hormone rhythms. The circadian rhythm itself is a deeply embedded biological system. It is not unique to humans. Virtually every organism that has been studied, from single-celled cyanobacteria to plants to insects to mammals, has some form of internal timing system that responds to the light-dark cycle. In mammals, the molecular machinery of the circadian clock involves a set of genes that interact in a feedback loop, cycling through a roughly 24-hour pattern of activation and suppression. The proteins produced by these genes regulate the timing of many other cellular processes throughout the body. In humans, the core clock genes include ones named clock, BMAL1, PER1, PERR2, CRY1, and CRY2. These genes interact in cycles that take roughly 24 hours to complete. Clock and BAM-AL1 proteins activate the production of PER and CRY proteins. As PER and CRY proteins accumulate, they begin to suppress the activity of CLOCK and BMAL1, slowing their own production. As PER and CRY levels fall, the suppression lifts and clock and BMAL1 become active again. This loop runs continuously in nearly every cell of the body. The SCN coordinates these individual cellular clocks across all the body's tissues using a combination of nerve signals, hormones, and body temperature rhythms. Light input via the IPRGCs is the primary external signal that keeps the system synchronized with the actual 24-hour day. The monthly variation in nighttime light caused by the lunar cycle means that the light environment that the body's timing systems are monitoring changes predictably over the course of each month. On nights near the new moon, nighttime light levels are at their lowest. So, nights near the full moon, they are at their highest, at least from natural sources. Whether this monthly variation in natural light produces detectable changes in melatonin levels, sleep timing, or circadian phase in modern humans living with artificial light is a specific research question. The answer is not fully resolved, but several studies have attempted to address it, with results that suggest the effect may be real, though small, even in people exposed to artificial light at night. Before electric lighting, the monthly cycle of moonlight would have been a much more prominent feature of the nighttime light environment. Nights near the full moon would have been noticeably brighter than nights near the new moon, and this difference would have been experienced every month without interruption throughout a person's lifetime. The visual and circadian systems that humans carry today evolved during this period when moonlight was the primary source of nighttime illumination. Whether the brain retains any functional sensitivity to the monthly light cycle that goes beyond simple response to absolute light levels is one of the more interesting open questions in chronobiology, the scientific field that studies biological timing systems. The physics of how moonlight reaches Earth's surface also includes some effects that are specific to the full moon phase. When the moon is full, sunlight strikes its surface at a nearly head-on angle, with very little shadow falling across the surface features. On any other night, the angle of sunlight creates shadows across craters, ridges, and rock formations. These shadows reduce the total amount of light reflected back toward an Earth. At full moon, with the sun directly behind the observer, as seen from the moon, almost all surface features are illuminated and shadows are minimized. The result is that the full moon is disproportionately bright compared to, for example, a half moon. The full moon is not just twice as bright as the half moon, it is actually about 9 to 10 times brighter. This nonlinear relationship between phase and brightness is a property of the moon's surface and is related to the way the microscopic structure of lunar regolith scatters light back toward its source, a phenomenon called the opposition surge or Heiligenschein effect in planetary science. This means the full moon delivers significantly more light energy to Earth's surface than the sum of the days immediately before and after it in the cycle. The peak brightness is concentrated around the 1 to 2 nights of the full moon itself, rather than being spread evenly across several nights. From a biological standpoint, this concentrated peak of nighttime light is what makes the full moon distinct from the rest of the lunar cycle as a potential environmental signal. Sleep is not a single state. It is a structured process that moves through several distinct stages in a predictable order, repeating in cycles throughout the night. Understanding what the full moon may or may not do to sleep requires first understanding what normal sleep looks like at a biological level, because the changes researchers have looked for are changes within this structure, not simply changes in whether a person sleeps or not. A typical night of sleep in a healthy adult it consists of 4 to 6 complete cycles, each lasting roughly 90 minutes. Each cycle moves through a sequence of stages. The first is a light stage of non-REM sleep, where the body begins to relax and brain activity starts to slow. This transitions into a deeper stage of non-REM sleep, and then into the deepest stage, called slow wave sleep. Slow wave sleep is the stage where physical restoration is most concentrated. Growth hormone is released in large amounts during this stage. The immune system carries out significant repair work. The body's energy stores are partially replenished. Brain activity during slow wave sleep is characterized by large, slow electrical waves that sweep across the cortex at a rate of about 1 per second. These are called delta waves, and the proportion of slow wave sleep in a night is one of the clearest indicators of sleep quality. After slow wave sleep, the cycle moves into REM sleep, which stands for rapid eye movement sleep. During REM, the brain becomes much more active, producing electrical patterns that look somewhat similar to wakefulness. The eyes move rapidly beneath closed lids, the muscles of the body, except for those controlling breathing and eye movement, become temporarily paralyzed. Most vivid dreaming occurs during REM sleep. REM sleep is associated with memory consolidation, emotional processing, and certain types of learning. The proportion of REM sleep increases across the night, meaning the later sleep cycles contain more REM than the earlier ones. Against this backdrop, researchers have asked a specific question. Does the phase of the moon change any of these measurable features of sleep? The first study to attract wide attention on this question was published in 2013 by a team led by Christian Kayachin at the University of Basel in Switzerland. The researchers analyzed sleep data that had been collected over several years in a controlled laboratory setting where participants slept in a room with no windows and no access to information about the time or the lunar phase. The data was analyzed retrospectively, meaning the researchers looked back at the records and compared sleep measurements taken during different phases of the lunar cycle. The results were notable. Around the full moon, participants took about 5 minutes longer to fall asleep. Their total sleep time was reduced by about 20 minutes on average. The amount of slow wave sleep decreased by about 30%, and melatonin levels measured in the blood were lower around the full moon than at other points in the cycle. The EEG readings, which measure electrical activity in the brain during sleep, showed reduced delta wave power during the full moon period, consistent with the reduction in slow wave sleep depth. The participants reported feeling less rested after sleep near the full moon, which aligned with the objective measurements showing reduced sleep quality. Because the participants had no windows and no way of knowing what phase the moon was in, the researchers argued that these changes could not be explained by psychological expectation or awareness of the full moon. The effect appeared to be present, even in the absence of any direct moonlight exposure or conscious knowledge of the lunar phase. This raised the possibility that humans might retain some internal biological response to the lunar cycle that operates independently of direct light exposure. The study was widely reported and generated significant scientific interest, but it also drew criticism. The sample size was relatively small, around 33 participants. The retrospective nature of the analysis meant the study was not originally designed to test lunar effects, which introduced the possibility of statistical artifacts. Some researchers pointed out that the effect, though statistically significant in the analysis, was not large in absolute terms. A 20-minute reduction in total sleep time and a 5-minute increase in sleep onset time are measurable but modest changes. Other scientists attempted to replicate the findings with larger groups and did not always find the same results. Subsequent studies produced a mixed picture. Some found small but consistent associations between the full moon and changes in sleep duration or timing. Others found no significant relationship. A large study published in 2021 using data from wrist-worn activity monitors worn by people in rural communities in Argentina, Bolivia, and the United States found that sleep onset was delayed by 30 to 80 minutes, and total sleep time was reduced by 45 to 90 minutes in the days leading up to a full moon. This study covered over 500 people across different environments and lighting conditions, and the effect was stronger in communities with less access to artificial light, suggesting that electric lighting may reduce, but not eliminate, a residual lunar effect on sleep timing. The consistency across communities with very different artificial light exposure was one of the more interesting aspects of this research. People in rural communities without electricity showed the largest shift in sleep timing around the full moon. People in urban environments showed smaller but still detectable shifts. This pattern is consistent with the idea that the lunar light cycle influences sleep, but that artificial light competes with and partially masks this influence in modern environments. What remains unresolved is the mechanism. If the full moon affects sleep through light exposure, then controlling for light, as the Basel study attempted to do, should eliminate the effect. The fact that the Basel study found an effect, even in the absence of moonlight, suggests either that the statistical analysis captured a real internal biological rhythm, or that the effect in that study was a statistical artifact that did not represent a true biological response. These two possibilities have not been definitively separated, and researchers continue to design studies aimed at resolving the question. Brainwave data collected during sleep near the full moon has been a consistent focus of this research. The reduction in delta wave power found in the Basel study points specifically to slow wave sleep being affected. Slow wave sleep is the stage most sensitive to disruption by light, noise, and other environmental factors. It is also the stage with the most direct links to physical health outcomes. Repeated reductions in slow wave sleep over time are associated with impaired immune function, slower physical recovery, and changes in metabolic regulation. Whether the modest changes observed in full moon studies are large enough to produce meaningful health effects over a lifetime is a question that has not been answered. The brain does not simply shut down during sleep. It shifts into a different mode of operation, one that is in many ways just as active as wakefulness, but directed toward entirely different tasks. The work the brain does during sleep is essential, and the conditions under which sleep occurs, including the level of light in the environment, directly affect how well that work gets done. During waking hours, the brain is constantly receiving and processing information. Neurons fire in rapid, complex patterns. Neurotransmitters are consumed. Metabolic waste products accumulate as a byproduct of all this activity. The brain uses about 20% of the body's total energy supply, despite accounting for only about 2% of body weight. This high level of activity produces cellular byproducts, including proteins that, if they accumulate in large amounts, can become damaging. One of the most studied of these is a protein called amyloid beta, which is associated with Alzheimer's disease when it builds up in abnormal deposits called plaques. For many years, scientists understood that the brain needed sleep, but were not entirely clear on one of the most basic reasons why. A major discovery in 2013 helped change that understanding. Researchers at the University of Rochester, working with mice, identified a previously unknown waste clearance system in the brain that operates primarily during sleep. They called it the glymphatic system. The name combines glial, referring to the brain's non-neuronal support cells called glia, and lymphatic, referring to the body's waste drainage system that operates in other tissues. The glymphatic system works through the spaces around blood vessels in the brain. Cerebrospinal fluid, a clear liquid that surrounds and cushions the brain and spinal cord, is pumped through these spaces in a directed flow. As it moves, it carries waste products, including amyloid beta and other metabolic byproducts, out of the brain tissue and toward drainage points, where they are eventually cleared from the central nervous system. This process happens continuously to some degree, but the 2013 research showed that during sleep, particularly during slow wave sleep, the flow rate of cerebrospinal fluid through the brain increases dramatically. The cells of the brain actually shrink slightly during sleep, expanding the spaces between them and allowing cerebrospinal fluid to move more freely. The researchers estimated that the clearance of waste products from the brain during sleep was about 10 times more efficient than during wakefulness. This finding reframed the question of why sleep is necessary. One major function of sleep, at least from the brain's perspective, appears to be maintenance, specifically the removal of metabolic waste that would otherwise accumulate and cause damage over time. Chronic sleep deprivation in this context is not just a matter of feeling tired. It potentially means that the brain's waste clearance system is not operating at full capacity on a regular basis, which could have long term consequences for brain health. The glymphatic system's dependence on slow wave sleep is relevant to the full moon research discussed earlier. If slow wave sleep is consistently reduced during full moon periods, even by modest amounts, and if the glymphatic system's efficiency is tied to slow wave sleep depth and duration, then a reduction in slow wave sleep would theoretically mean reduced brain waste clearance on those nights. Whether the scale of the reduction observed in full moon studies is large enough to produce any measurable difference in glymphatic function is not yet known. The link between lunar phase, sleep quality, and glymphatic activity has not been directly studied, but the mechanistic pathway is coherent enough that it represents a legitimate area for future research. Memory consolidation is another major function of sleep, and it too is tied to specific sleep stages. The relationship between sleep and memory involves two broad categories of memory. The first is declarative memory, which covers facts and events, the kind of information that can be consciously recalled and described. The second is procedural memory, which covers skills and habits, the kind of learning that is expressed through performance rather than conscious recall. Declarative memory consolidation is strongly associated with slow wave sleep and with a specific pattern of brain activity called sleep spindles. Sleep spindles are brief bursts of electrical activity in the brain, lasting about half a second to 2 seconds, that occur repeatedly during non REM sleep. They appear as sudden increases in the frequency of brain waves visible on EEG recordings. A typical night of sleep contains hundreds of sleep spindles. They are generated by a structure in the brain called the thalamus, which acts as a relay station for sensory information and plays a central role in coordinating communication between different brain regions during sleep. During slow wave sleep, the hippocampus, a brain region critical for forming new memories, replays patterns of activity that occurred during the day. This replay is thought to gradually transfer information from the hippocampus, where memories are initially stored in a relatively fragile form, to the cortex, where they are stored in a more stable long-term form. Sleep spindles appear to play a role in this transfer, helping to coordinate the timing of communication between the hippocampus and cortex. Studies that have artificially induced more sleep spindles using mild electrical stimulation during sleep have found improvements in declarative memory performance the next day. Procedural memory, by contrast, is more closely associated with REM sleep. Motor skills, musical performance, and certain types of pattern recognition all show improvement after a night of sleep, and this improvement appears to be particularly linked to the amount of REM sleep obtained. The brain during REM sleep shows activity in motor areas and in regions involved in processing visual and spatial information. It is as though the brain is rehearsing the movements and sequences learned during the day, consolidating them into more automatic and reliable patterns. The practical implication is that both types of memory depend on a complete and undisturbed night of sleep containing adequate amounts of both slow wave and REM sleep. Disruptions to either stage affect the consolidation of different types of information. A night with reduced slow wave sleep may impair the consolidation of factual memories learned the previous day. A night with disrupted REM sleep may slow the refinement of motor skills or procedural learning. Light plays a direct role in regulating the timing and structure of sleep through its effects on the circadian clock. The SCN, which receives light information from the retina via the IPRGCs, coordinates the timing of sleep by regulating body temperature and melatonin release. Body temperature follows a clear 24-hour rhythm. It rises through the morning and afternoon, peaks in the early evening, and then begins to fall in the hours before sleep. The drop in body temperature is a signal to the brain that sleep is approaching, and it continues to fall through the first half of the night, reaching its lowest point in the early morning hours before rising again toward waking. This temperature rhythm is tightly coupled to the light dark cycle. Exposure to bright light in the evening delays the temperature drop and shifts the timing of sleep later. Exposure to light in the early morning advances the temperature rhythm and shifts sleep timing earlier. The timing of melatonin release follows a similar pattern. Together, these two signals, body temperature and melatonin, function as internal cues that prepare the brain and body for sleep at a specific time each night. When light exposure is irregular or occurs at the wrong time of day, these signals become misaligned. The brain may receive conflicting information, some signals indicating it is daytime and others indicating it is nighttime. This misalignment, called circadian disruption, is associated with a range of health consequences, including impaired immune function, metabolic changes, mood disturbances, and increased risk of certain chronic conditions. Shift workers, who regularly work at night and sleep during the day, experience chronic circadian disruption and show elevated rates of several health problems as a result. The sensitivity of the circadian system to light is highest during two windows of the day. The first is in the morning, roughly in the first 2 hours after waking, when bright light exposure most effectively advances the clock and promotes alertness. The second is in the evening, in the 1 to 2 hours before the natural sleep time, when light exposure most effectively delays the clock and makes it harder to fall asleep at the usual time. Moonlight exposure falls in the evening and nighttime hours, which is precisely the window when the circadian system is most sensitive to light that could delay sleep timing. Even at low intensities, natural light during this sensitive window has the potential to interact with the circadian system, though the degree of this interaction under moonlight specifically has not been precisely quantified in human subjects. The brain's response to darkness is as active as its response to light. Darkness is not simply the absence of input. It is a specific condition that triggers a cascade of biological events. As evening light fades, the SCN begins reducing its suppression of the pineal gland, allowing melatonin production to begin. The brain's arousal systems, which are driven by neurotransmitters like norepinephrine, serotonin, and histamine, begin to reduce their activity. The adenosine system, which tracks how long the brain has been awake and generates increasing pressure to sleep, reaches a point where its signals become difficult to override. The brain begins to prepare for sleep not as a passive process of simply becoming less active, but as an active transition managed by specific neural circuits. The ventrolateral preoptic area, a small cluster of neurons in the hypothalamus, plays a central role in this transition. When activated, it suppresses the brain's arousal systems, essentially putting the brakes on wakefulness. It is mutually inhibitory with the arousal systems, meaning each side suppresses the other. This creates a kind of switch that tends to be in one of two stable states, either strongly awake or strongly asleep, with rapid transitions between them. Light information reaching the SCN influences the timing and stability of this switch. Bright light keeps the arousal systems active and the ventrolateral preoptic area suppressed. Darkness allows the switch to flip towards sleep. The electrical activity of the brain changes dramatically as this transition occurs. In the waking state, EEG recordings show high frequency, low amplitude waves, a pattern called beta and gamma activity, reflecting the rapid and varied firing of large numbers of neurons processing diverse streams of information. As the brain moves towards sleep, this activity slows. Alpha waves emerge at a frequency of about 8 to 12 cycles per second, associated with relaxed wakefulness and closed eyes. As light sleep begins, these give way to theta waves, slower still at 4 to 8 cycles per second. Then come the sleep spindles and K complexes of stage 2 non REM sleep, followed by the large, slow delta waves of slow wave sleep at less than 4 cycles per second. Each of these stages of electrical activity reflects a different state of brain organization. In slow wave sleep, large populations of neurons fire together in slow, synchronized waves that sweep across the cortex. Between these waves, there are brief periods of near silence when neurons are in a state of reduced activity called the down state. This alternation between active up states and silent down states is the hallmark of slow wave sleep and appears to be essential for the memory consolidation and waste clearance processes that depend on this stage. REM sleep, which follows slow wave sleep in the sleep cycle, shows a very different electrical pattern. The brain during REM produces fast, irregular waves that resemble waking activity, but the source and organization of this activity is different. The prefrontal cortex, which is the region most associated with rational thought, planning, and self monitoring, is relatively less active during REM compared to waking. At the same time, areas involved in emotion, visual processing, and autobiographical memory are more active. This combination produces the vivid, emotionally charged, and sometimes illogical experiences of dreaming. The brainstem plays a central role in generating REM sleep. A group of neurons in the brainstem, called the REM on cells, become active and trigger the characteristic features of REM, including the rapid eye movements, the suppression of muscle tone, and the activation of the cortex in the REM pattern. Another group, the REM off cells, suppresses REM during non REM sleep and waking. The interaction between these two groups determines when the brain enters and exits REM sleep throughout the night. The entire architecture of sleep, from the initial transition through slow wave sleep and REM cycles back to lighter stages before the next cycle begins, is sensitive to environmental conditions. Temperature, noise, stress, and light all affect the depth, timing, and completeness of each stage. Light in particular, because of its direct line to the SCN and the circadian timing system, has a privileged role in shaping when sleep begins, how deep it goes, and when it ends. The natural monthly variation in nighttime light from the lunar cycle means that the brain's sleep systems are operating against a slightly different background of environmental light from one week to the next, repeating on a 29.5-day schedule that has been a constant feature of the nighttime environment throughout human evolutionary history. The human body runs on cycles. Some last hours, some last days, and some stretch across weeks. These cycles are not independent of each other. They share common regulatory systems, respond to many of the same environmental signals, and interact in ways that researchers are still working to fully understand. The monthly lunar cycle, with its predictable shift in nighttime light, sits alongside several of the body's own monthly rhythms in a relationship that has been observed and studied for decades without being fully resolved. The menstrual cycle is the most discussed of these in relation to the moon. The average length of the menstrual cycle in humans is approximately 29.5 days, which is nearly identical to the length of the lunar synodic month. This numerical coincidence has attracted attention for a long time, from ancient observers who noticed it to modern researchers who have attempted to determine whether the similarity reflects a genuine biological connection or simply a coincidence of timing. The question is not simple, and the evidence does not point cleanly in one direction. A study published in 2021 in the journal Science Advances analyzed menstrual cycle records from 22 women over periods ranging from 1 to over 3 years. The researchers found that in women with cycles longer than 27 days, The timing of menstruation showed a tendency to synchronize with specific lunar phases over time, but this synchronization was intermittent rather than constant. Younger women showed stronger synchronization with the full moon and new moon than older women. The researchers suggested that modern artificial light, which is present at relatively constant levels regardless of lunar phase, may disrupt any natural tendency for the menstrual cycle to align with the lunar cycle, and that this disruption may become more pronounced with age and cumulative exposure to artificial light environments. Earlier work on this topic, including a large study by Winifred Cutler published in 1980, found a statistical tendency for menstruation to cluster around the new moon phase. Cutler's work was conducted before the widespread study of chronobiology as a field, and then the methods used have been debated in subsequent literature. More recent analyses using larger datasets, including a 2013 study that analyzed over 7.5 million cycles from an app-based dataset, found no significant synchronization between menstrual timing and lunar phase across the full population. The discrepancy between these findings reflects the difficulty of studying this question at scale, where individual variation, age differences, geographic differences in light exposure, and the influence of artificial light all introduce noise into the data. What is clear is that the menstrual cycle is regulated by a hormonal system that is sensitive to light. The hypothalamus releases a hormone called gonadotropin-releasing hormone in pulses. These pulses trigger the pituitary gland to release two other hormones, luteinizing hormone and follicle-stimulating hormone, which in turn regulate the ovarian cycle, the development and release of eggs, and the production of estrogen and progesterone. The timing of these hormonal pulses is influenced by the circadian clock in the SCN, which means that anything affecting circadian timing, including light exposure patterns, can in principle affect the hormonal regulation of the menstrual cycle. Melatonin has a direct inhibitory effect on the release of gonadotropin-releasing hormone. Higher melatonin levels, which occur during longer nights in winter or in conditions of low artificial light exposure, suppress the reproductive hormone axis to some degree. Lower melatonin levels, as occur during summer or under higher nighttime light conditions, allow the reproductive hormone axis to operate with less suppression. This relationship is most clearly expressed in seasonally breeding animals, where the length of night tracked through melatonin duration determines whether the animal enters a reproductive state. In humans, seasonal effects on reproduction are subtle but detectable in population-level birth rate data, which shows small but consistent seasonal patterns in many parts of the world. Cortisol is another hormone that interacts closely with light and the circadian system. It is produced by the adrenal glands and follows a strong 24-hour rhythm. Cortisol levels are at their lowest during the middle of the night and begin rising in the hours before waking, reaching a peak in the first 30 to 45 minutes after waking in what is called the cortisol awakening response. This morning peak prepares the body for the demands of the day. It raises blood sugar, increases alertness, and mobilizes energy stores. Throughout the day, cortisol levels gradually decline, reaching low levels again by evening. The timing of this cortisol rhythm is set by the circadian clock. Light exposure in the morning is one of the primary signals that calibrates the cortisol awakening response. People who waken to bright light show a stronger morning cortisol peak than those who wake in darkness. Disruptions of the light-dark cycle, such as those experienced by shift workers or people with irregular sleep schedules, alter the timing and amplitude of the cortisol rhythm. Chronically disrupted cortisol patterns are associated with impaired immune function, increased inflammation, and changes in mood and cognitive performance. The interaction between moonlight, melatonin, and cortisol over the course of the monthly cycle has not been studied in detail. What is known is that the hormonal systems involved are sensitive to light, that light exposure patterns affect both melatonin and cortisol timing, and that the full moon represents the brightest point in the natural monthly light cycle. Whether the modest increase in nighttime light around the full moon is sufficient to produce detectable shifts in hormone levels. in people living under modern conditions is a question that requires more targeted research than has yet been conducted. What the existing evidence does suggest is that the body's hormonal systems did not evolve in isolation from the lunar light cycle. For most of human evolutionary history, the full moon was the brightest source of nighttime light available. The monthly rhythm of melatonin suppression that this light cycle would have produced, even modestly, would have been a regular feature of the hormonal environment. Whether the body retains calibrated sensitivity to this rhythm, or whether it has been effectively overridden by the constant presence of artificial light, remains one of the genuinely open questions in the study of human chronobiology. The natural world does not treat the full moon as an ordinary night. Across an enormous range of species, from animals living deep in tidal zones to those moving through open grasslands, the monthly peak of nighttime light triggers measurable changes in behavior. These changes have been documented by researchers working across marine biology, entomology, ornithology, and mammalian ecology. They are not uniform in their character. Different species respond in different ways, and the mechanisms driving these responses vary. But the pattern that emerges across all of this research is consistent. The full moon is an environmental event that living systems notice and respond to. The ocean provides some of the most dramatic and well documented examples of lunar driven behavior. The waters around coral reefs are home to one of the most precise biological timekeeping events known in nature. Many species of coral reproduce through a process called broadcast spawning, in which large numbers of individual coral colonies release their eggs and sperm into the water simultaneously. The mass release creates a visible phenomenon, sometimes described as an underwater snowstorm, as clouds of reproductive material rise through the water. For spawning to be effective, the release must be synchronized across enormous numbers of individuals spread across wide areas of reef. If individual colonies release their gametes at different times, the chances of eggs meeting sperm from the same species would be low. Synchronization is not just useful, but it is essential. On the Great Barrier Reef off the coast of Australia, mass coral spawning occurs once a year, in the spring, in the days immediately following the full moon of October or November. The specific timing involves several environmental cues working together. Water temperature must have risen to a threshold level consistent with the spring season. The length of the day must be within a specific range, and the lunar phase must be at or just past full. When all three conditions are met simultaneously, spawning begins, typically on the third to fifth night after the full moon, usually within a narrow window of 1 to several hours after sunset. The precision of this timing is remarkable. Researchers studying the neural and hormonal mechanisms behind coral spawning have identified light-sensitive proteins in coral tissue that appear to function as lunar light detectors. These proteins, called cryptochromes and opsins, respond to the specific quality of moonlight at different phases of the lunar cycle. The blue-green wavelengths that moonlight contains at full phase appear to be particularly important. As the full moon approaches, the light signal reaching the coral tissue changes in intensity and spectral character, and this change appears to trigger the final hormonal cascade that leads to spawning. The precise molecular pathway is still being investigated, but the sensitivity of coral tissue to lunar light has been confirmed across multiple studies. The Christmas Island red crabs provides another striking example of lunar-timed reproductive behavior in a marine context. These land crabs, which live in the rainforests of Christmas Island in the Indian Ocean, migrate from their inland forest habitat to the coast each year to release their eggs into the ocean. The migration is timed to the lunar cycle with high precision. The female crabs release their eggs at the shoreline during the last quarter moon phase, when tidal conditions create a particular flow of water near the shore that carries the eggs out to sea. The migration begins several weeks before this release, with the crabs sensing both seasonal cues and lunar phase cues to time their departure from the forest. The total population involved in this migration numbers in the tens of millions, and the synchronization of their movement and reproductive release is driven substantially by lunar timing. Marine invertebrates more broadly show a wide range of lunar timed behaviors. Bristleworms of the genus Platinaris reproduce in coordinated mass events around the full moon and new moon, with the timing controlled by a circulunar clock, an internal biological timer that runs on approximately a monthly cycle. This internal clock has been studied in laboratory conditions where worms are kept under constant artificial light, removing any direct moonlight cue. Even in these conditions, the worms maintain a roughly monthly reproductive rhythm for several months before the clock gradually drifts out of phase. This demonstrates that the lunar timing is not purely driven by external moonlight detection, but involves a genuine internal oscillator, analogous to the circadian clock, but running on a monthly rather than daily period. The molecular basis of the circulunar clock has been an active area of research. Studies on Platinaris worms have found that some of the same genes involved in the circadian clock, including cryptochrome genes, also play a role in the circulunar clock, though the exact mechanism and how the two clocks interact is not yet fully understood. The discovery of a genuine internal monthly clock in marine invertebrates has raised the possibility that similar circulunar clocks may exist in other animals, including vertebrates, though the evidence for this in mammals and birds is less direct. Fish behavior under the full moon has been documented across many species and environments. Coral reef fish show increased activity and altered movement patterns during full moon nights. Predatory species that hunt by sight benefit from the increased nighttime illumination and extend their active hunting hours. Prey species correspondingly alter their behavior, spending more time in sheltered areas and reducing their exposure in open water. The interplay between predator and prey activity patterns around the full moon creates a shift in the ecology of the reef that researchers have described as a monthly reorganization of the community's behavior. Grunion, a small fish found along the Southern California coast, perform one of the most precisely timed reproductive events of any vertebrate. On specific nights following the full and new moons of spring and summer, Grunion ride breaking waves onto the beach. Females partially bury themselves in the wet sand and deposit their eggs. Males curve around the females and release sperm, which runs down the female's body and into the sand to fertilize the eggs. The whole process is completed in a matter of minutes before the fish ride the next wave back into the ocean. The eggs are left in the sand above the waterline, where they develop over the next 2 weeks. The timing of their hatching is also tied to the tidal cycle. The next series of high tides following the new or full moon reaches up the beach, agitates the eggs, and triggers hatching, sending the larvae into the surf. The entire reproductive sequence, from spawning to hatching, is calibrated to the lunar tidal cycle with a precision that leaves almost no room for error. Insects respond to lunar light in ways that reflect both the opportunities and dangers that full moonlight creates. Moths, which are primarily nocturnal, use the moon as a navigational reference. The traditional understanding was that moths flew in straight lines by keeping a constant angle to the moon, a strategy that works well over short distances but causes the moth to spiral inward when the light source is very close, which is why moths appear to circle artificial lights. Research has added considerable nuance to this picture. Different moth species show different responses to lunar illumination. Some species reduce their flight activity during full moon nights, apparently to reduce their visibility to bat predators that hunt by echolocation but also use vision when light allows. Other species increase activity during full moon nights, taking advantage of the improved navigation conditions. Dung beetles, which feed and reproduce using animal dung, use the moon as a navigational tool in a precise and documented way. Researchers studying the African dung beetle Scarabaeus satyrus found that these animals can orient and navigate using the pattern of polarized light from the moon. On full moon nights, dung beetles roll their dung balls in straight lines away from the dung pile, using the moon's position to maintain direction. On overcast nights or nights with no moon, navigation becomes less precise and the paths more curved. The beetles can also use the Milky Way as a navigational reference under moonless conditions, but the moon is the primary cue when available. This represents one of the only documented cases of an insect navigating using moonlight as a compass. Mosquito activity is affected by lunar phase in ways that have practical public health implications. Several studies across different geographic regions and mosquito species have found that mosquito biting rates and activity levels change across the lunar cycle, with some species showing peaks near the full moon and others showing reduced activity. The mechanisms appear to involve both light driven activity patterns and lunar timed changes in the mosquito's internal cycles. In regions where mosquitoes transmit malaria and other diseases, understanding these lunar activity patterns could in principle be useful for timing interventions, though this remains a largely theoretical application. Bird behavior under the full moon reflects a combination of light driven activity and the potential for lunar timing to affect migration. Many bird species migrate at night, navigating using stars, magnetic fields, and potentially the moon. Radar studies tracking nocturnal bird migration have found that migration intensity, the number of birds in the air on a given night, is correlated with lunar phase in some regions and species. Migration tends to be more intense on bright moonlit nights in some studies, less intense in others, with the direction of the effect apparently depending on the species and the specific navigational challenge of the route. The full moon appears to reduce the risk of certain predators for migrating birds in some environments, since the bright sky makes it easier for birds to detect and avoid aerial predators. Shorebirds that feed in tidal zones adjust their feeding schedules to take advantage of low tide, which exposes mudflats and invertebrate prey. Since tidal timing shifts with the lunar cycle, the feeding schedules of these birds shift accordingly. Around the full and new moon, when spring tides occur and the tidal range is largest, more intertidal area is exposed during low tide, providing more feeding opportunity. Shorebirds have been observed spending more time feeding during these periods, extending their activity into nighttime hours when the full moon provides enough light for continued foraging. Among mammals, the responses to full moon illumination are varied and in many cases directly related to predator-prey dynamics. The increased light of the full moon makes it easier for visually oriented predators to detect and pursue prey. Many prey species have evolved behavioral responses that reduce their exposure during full moon periods. Studies on small rodents have found that their movements are more restricted during full moon nights, with animals spending more time in dense cover and less time in open areas compared to darker nights. This pattern has been documented in multiple species across different continents, suggesting it reflects a broadly conserved response to elevated predation risk under bright nighttime conditions. Lion hunting behavior provides a well-documented example from large mammal ecology. Lions are capable hunters in low light, but become significantly more effective when visibility is higher. Analysis of lion attack records from the Serengeti in Tanzania found that lion predation rates on humans were highest in the nights immediately following the full moon, when the moon rises late and the early evening hours are darker than on the night of the full moon itself. In contrast, human fatalities from lion attacks were lowest in the nights immediately after the new moon, when darkness prevails all night. The researchers noted that lions appeared to capitalize on the dark first half of the night following the full moon, when they had spent the previous days in conditions of high moonlight illumination and may have been experiencing a kind of predatory readiness or hunger buildup from reduced hunting success during the bright full moon nights. This study illustrated how lunar-driven changes in light create cascading effects through the predator-prey system over the course of each monthly cycle. Bats, which hunt primarily using echolocation rather than vision, show a different pattern. Since they do not rely on vision for prey detection, bright moonlight offers them less of an advantage in hunting. However, it does increase their own visibility to potential predators. Some bat species show reduced flight activity during full moon nights, a behavior termed lunar phobia in the research literature. Others show no significant change. The response appears to vary by species based on their size, habitat, and the predators they face. Smaller bats, which are more vulnerable to owl predation, show stronger lunar phobia than larger species. This suggests the behavioral response is tuned to the actual predation risk faced by each species rather than being a generic response to moonlight. The consistency of lunar driven behavioral responses across such a wide range of animal types points to something fundamental about the relationship between living systems as in the monthly light cycle. The mechanisms vary enormously. Some species use internal circulunar clocks that operate independently of direct moonlight exposure. Others respond directly to the light intensity of each night. Many use a combination of both. The behaviors triggered by these mechanisms range from reproduction to migration to foraging to predator avoidance. But across all of this diversity, the full moon emerges as a consistent and meaningful point in the monthly cycle, a peak of environmental light that has been shaping the behavior of living systems for far longer than any of the individual species responding to it have existed. Long before any written record existed, humans were watching the moon. The evidence for this is physical. Bones and stones carved with sequences of notches, some dating back 30,000 years or more, have been found at archaeological sites across Europe and Africa. The patterns of these notches correspond in some cases to the number of days in a lunar cycle, or to multiples of it. Whether these objects functioned as calendars in a formal sense is debated among researchers, but they demonstrate that people were tracking the moon's cycle with enough consistency to record it in a durable material. The moon was not simply observed, it was counted. The Ashango bone, found in what is now the Democratic Republic of Congo and dated to roughly 20,000 years ago, carries 3 columns of notches, whose groupings that some researchers have interpreted as a record of lunar phases across several months. The interpretation is not universally accepted, and the exact function of the bone remains uncertain. But the mathematical structure of the notch groups is difficult to explain as accidental, and the lunar calendar interpretation is one of the more coherent explanations available. Similar marked bones and antlers from sites in France and Spain suggest that this kind of systematic moon tracking was widespread across different human populations during the Upper Paleolithic period. The transition from simple notch counting to formal calendrical systems happened gradually as human societies became more complex. Among the earliest well-documented lunar calendars is the one used in ancient Mesopotamia, the region between the Tigris and Euphrates rivers, in what is now Iraq. Sumerian and later Babylonian civilizations built their calendar around the lunar months. Each month began with the first visible crescent of the new moon, observed by trained watchers stationed at elevated points. The month lasted until the next crescent appeared, giving months of either 29 or 30 days, depending on when the crescent became visible. 12 such months gave a year of about 354 days, roughly 11 days shorter than the solar year of 365.25 days. This difference between the lunar year and the solar year created a problem for any civilization that needed to track both the moon and the agricultural seasons, which are determined by Earth's position in its orbit around the sun rather than by the lunar cycle. If the calendar simply followed 12 lunar months, it would drift backward relative to the seasons by about 11 days each year. Over 3 years, this drift would amount to about 33 days, more than a full month. Planting seasons, flood cycles, and harvest times, all of which depend on the solar year, would no longer align with the calendar months that they were supposed to govern. The Babylonians solved this problem by adding an extra month to the calendar periodically, a practice called intercalation. By inserting a 13th month in certain years, they kept the lunar calendar roughly aligned with the solar year over longer periods. This was initially done on an ad hoc basis, decided by royal decree when the drift became noticeable. Over time, Babylonian astronomers developed a more systematic approach. By the 5th century BCE, they had worked out a 19-year cycle, now called the Metonic cycle, after the Greek astronomer Meton of Athens, who described it independently around 432 BCE, in which 235 lunar months fit almost exactly into 19 solar years. This cycle allows a lunar calendar to stay within a few days of the solar calendar over 19 years by adding 7 extra months distributed across the cycle in a specific pattern. The precision of Babylonian astronomical observation was remarkable for its time. Clay tablets from Babylon carry records of lunar and planetary positions extending back centuries. These records allowed Babylonian astronomers to identify repeating patterns in celestial events, predict lunar eclipses with considerable accuracy, and develop mathematical models of the moon's variable speed in its orbit. The recognition that the moon moves faster when closer to Earth and slower when farther away, a consequence of the elliptical shape of its orbit, was encoded in Babylonian mathematical astronomy, well before it was given a geometric explanation by later Greek thinkers. Ancient Egyptian timekeeping took a different approach. Egyptian civilization oriented its calendar primarily around the solar year and the annual flooding of the Nile, which was tied to Earth's position in its orbit. The Egyptian civil calendar used 365 days divided into 12 months of 30 days each, with 5 additional days added at the end of the year. This was one of the earliest solar calendars in recorded history. However, the moon was not ignored in Egyptian religious and ceremonial life. Lunar months governed the timing of many religious festivals, and a separate lunar calendar ran alongside the civil calendar for religious purposes. The two systems were maintained simultaneously, with priests responsible for tracking both. In China, the lunar calendar has been in continuous use for thousands of years, though in a form that incorporates solar year corrections similar to the Babylonian intercalation system. The traditional Chinese calendar is lunisolar, meaning it tracks lunar months but adds extra months periodically to keep the calendar aligned with the solar year. The timing of major traditional festivals, including the Lunar New Year and the Mid-Autumn Festival, is determined by this lunisolar calendar. The Mid-Autumn Festival falls on the 15th day of the 8th lunar month, which corresponds to the full moon of that month, and the celebration centers explicitly on the full moon as its defining astronomical event. The relationship between the lunar calendar and agriculture was not abstract. In many ancient societies, the moon was a practical farming tool. The visibility of the moon at night allowed farmers and laborers to extend their working hours during critical periods, such as planting and harvest. Fields could be worked by moonlight when the full moon provided enough illumination to see clearly. The predictability of the full moon's timing meant that extra labor during the brightest nights of the month could be planned in advance. In English, the full moon closest to the autumn equinox is still called the harvest moon, a name that reflects the historical agricultural importance of the extra evening light it provided during harvest season. Many ancient cultures also developed beliefs about how the phase of the moon affected the growth of crops, the best times for planting, and the behavior of animals. These beliefs varied considerably from one culture to another and were not always consistent with each other. Some held that planting should occur during the waxing moon to encourage growth. Others specified different phases for different types of crops. While modern agricultural science does not support most of these specific beliefs as causal relationships, they reflect the depth to which lunar observation was integrated into everyday decision-making across a wide range of ancient societies. The moon was not a background feature of the sky. It was a practical reference point, a timer, a calendar, and in many traditions, a governing force whose phases structured the rhythm of daily and seasonal life. The field of chronobiology, the scientific study of biological timing systems, has grown considerably over the past 3 decades. Advances in genetics, brain imaging, hormone measurement, and wearable monitoring technology have given researchers tools that were not available to earlier generations of scientists. These tools have made it possible to study the relationship between the lunar cycle and human biology with a level of precision that older studies could not achieve. The results have been informative, yet they have also revealed how much complexity lies beneath what initially seems like a straightforward question. The central question that modern research has tried to answer is whether the full moon produces measurable changes in human biology and behavior, and if so, through what mechanism. Answering this question turns out to require careful attention to study design, because the effects being investigated are modest in scale. The variables involved are numerous, and the history of this topic includes a significant amount of research that was conducted without the methodological rigor needed to produce reliable conclusions. Early studies on lunar effects in humans often relied on hospital records, police reports, emergency room admission data, and psychiatric facility records. Researchers would count events such as hospital admissions, violent incidents, or psychiatric crises, and compare the numbers across different lunar phases. Many of these studies found apparent correlations. Others found nothing. A major review published in 1985 by researchers James Rotten and Ivan Kelly analyzed 37 studies on lunar effects on human behavior and found that the studies claiming to find lunar effects suffered from significant methodological problems. These included small sample sizes, failure to control for confounding variables such as day of the week or seasonal effects, selective reporting of positive results, and inconsistent definitions of lunar phase boundaries. When these problems were accounted for, the apparent correlations largely disappeared. This critical review was important because it pushed the field toward higher standards. But it did not settle the question. What it showed was that the methods used in earlier research were not adequate to detect small, real effects if they existed, and were also not adequate to reliably rule them out. The tools available at the time simply were not precise enough. Counting emergency room admissions by lunar phase, for example, is a very blunt instrument. Emergency room visits are influenced by dozens of factors, including day of the week, season, weather, local events, and the availability of other healthcare options. A genuine lunar effect on sleep quality or hormone levels, if it existed, might produce changes too subtle to show up clearly in such noisy data. The 2013 Basel Sleep Study represented a shift in methodology. Rather than looking at broad behavioral outcomes, it examined specific physiological measurements taken in a controlled setting. EEG recordings, melatonin blood levels, and subjective sleep quality ratings gave a more granular picture than hospital admission counts. The controlled laboratory environment eliminated several confounders, particularly direct moonlight exposure and awareness of lunar phase. The findings showing reduced slow wave sleep, lower melatonin, and longer sleep onset time around the full moon were treated cautiously by the researchers themselves, who acknowledged that the retrospective nature of the analysis and the small sample size limited the strength of the conclusions. Subsequent attempts to replicate the Basel findings produced mixed results. A study published in 2014 by researchers at the Max Planck Institute for Psychiatry used a similar approach, but found no significant effect of lunar phase on sleep as measured by wrist actigraphy in a large sample of people monitored over extended periods in their home environments. The difference in findings between this study and the Basel study may reflect genuine differences between laboratory-controlled sleep and sleep in natural home environments, or it may reflect the statistical variability inherent in studying small effects with limited sample sizes. A 2021 study took a broader approach by collecting wrist actigraphy data from people in 3 communities, with very different levels of artificial light exposure. One community was in an urban area of Seattle in the United States with high artificial light exposure. Two communities were in rural Argentina, one with access to electricity and one without. The study tracked sleep timing and duration in relation to lunar phase across these groups. The results showed that sleep onset was delayed in the days leading up to the full moon in all three communities, but the effect was largest in the community without electricity and smallest in the urban community. Total sleep time was also reduced in the nights before the full moon. The researchers interpreted this as evidence that the lunar light cycle influences sleep timing and that artificial light reduces but does not eliminate this effect. This study was notable for several reasons. It used objective measurement rather than self report. It covered a range of light environments rather than a single setting, and it found a consistent directional effect across groups that differed substantially in their daily light exposure. The finding that the effect was strongest in the community without electricity is consistent with the hypothesis that the full moon's influence on sleep operates through light, since that community would experience the most dramatic difference in nighttime light between full moon and new moon nights. However, the study also found some effect in the urban community, where the difference in nighttime light between full and new moon would be largely masked by artificial light. This residual effect in the urban sample raises the possibility that some component of the lunar sleep effect may operate through a mechanism other than direct light exposure. One candidate mechanism that researchers have proposed is a genuine internal circulunar rhythm, analogous to the circadian clock but running on an approximately monthly period. The evidence for such a clock in marine invertebrates is well established, as discussed in the context of bristleworm reproductive timing. The evidence in humans is much less direct. No specific circulunar clock genes have been identified in humans, and no internal monthly oscillator has been directly demonstrated in human physiology. What exists is circumstantial evidence, including the apparent persistence of sleep related effects, even in controlled conditions, without direct moonlight exposure, and the numerical similarity between the average menstrual cycle length and the lunar month. These observations are consistent with the existence of a human circulunar clock, but they do not prove it. The methodological challenge of studying a circulunar clock in humans is significant. To demonstrate that an internal monthly oscillator exists, researchers would need to show that some biological rhythm persists on approximately a monthly period, even when all external monthly cues, including moonlight, tidal information, and any other environmental signal that cycles monthly, are removed. This is far more difficult to achieve than the analogous demonstration for the circadian clock, where people can be kept in constant conditions for days or weeks and still show clear 24-hour rhythms in temperature, hormone levels, and alertness. Keeping people in constant conditions for the months that would be needed to demonstrate a circulunar clock would be logistically and ethically difficult and has not been done. Wearable technology has opened new possibilities for studying lunar effects at scale. Devices that continuously monitor heart rate, movement, skin temperature, and sleep staging can collect data from large numbers of people over extended periods without requiring laboratory visits or controlled conditions. Several research groups have begun mining large data sets from these devices to look for lunar phase signals in physiological data. The challenge with this approach is that wearable data collected in free living conditions contains enormous amounts of variability from individual lifestyle differences, occupational patterns, travel, illness, and other factors. Statistical methods capable of extracting a small monthly signal from this background noise are still being developed and validated. Genetic research has contributed a new angle to the investigation. Studies in model organisms, particularly the marine worm Platynurus dumorilli, have identified specific genes involved in the circulunar clock, and some of these genes have counterparts in the human genome. This does not mean that humans have an active circulunar clock controlled by these genes, but it raises the possibility that the genetic machinery, for one, may be present in humans in a latent or modified form. Whether these genes play any role in human monthly rhythms is not currently known, and investigating this question requires both molecular biological tools and the ability to study human physiology across multiple monthly cycles under carefully controlled conditions. The question of artificial light as a confounding factor runs through all modern research on lunar effects in humans. Electric lighting has been present in human environments for roughly 150 years. Before that, the full moon was by far the brightest source of nighttime light available. The biological systems that regulate sleep, hormone production, and circadian timing evolved over hundreds of thousands of years in an environment where the monthly light cycle of the moon was a constant and prominent feature of the nighttime sky. If these systems retain any calibrated sensitivity to the lunar light cycle, the sensitivity may now be chronically overridden by artificial light, making it difficult to detect in modern populations. Researchers studying populations with low artificial light exposure, such as the indigenous communities in the 2021 actigraphy study, are trying to use this natural variation in light environments to shed light on this problem. The logic is that if lunar effects on sleep are larger in low light communities, this supports the hypothesis that the effects are light mediated and that artificial light suppresses them. But even communities with no access to electric lighting still live in a world with many other monthly variables, including social and economic cycles that may affect sleep and behavior independently of the moon. The neuroscience of how the brain might respond to monthly light variation has received less direct attention than the epidemiological and chronobiological aspects of the lunar research. Brain imaging studies looking at neural activity in relation to lunar phase have been limited in number and scope. One area of interest is the default mode network, a set of brain regions that are active during rest and inwardly directed thought, and that show altered activity patterns in relation to circadian phase. Whether these networks also show monthly modulation in relation to lunar phase has not been systematically studied. Another area of interest is the amygdala, a brain structure involved in processing emotional significance and threat, which has been proposed as a potential site for lunar modulation of behavior based on its sensitivity to environmental light signals. But direct evidence for this is limited. The psychiatric literature has a long history of claimed associations between the full moon and changes in mood, psychosis, and emergency psychiatric presentations. These claims have been studied extensively, and the results have been largely negative. Large analyses of psychiatric hospital admissions, police records of violent incidents, and emergency department data have generally found no consistent lunar phase effect once methodological controls are applied. The persistence of the popular belief that the full moon affects mental health, despite the lack of strong supporting evidence, is itself a subject of scientific interest. Researchers have proposed that confirmation bias, the tendency to notice and remember events that confirm existing beliefs while overlooking those that do not, plays a significant role in maintaining this belief in both the general public and among some healthcare workers. The open questions in this field are genuine and substantial. Does a human circulunar clock exist? If so, what genes are responsible, and how does it interact with the circadian system? Is the sleep effect observed in some studies mediated entirely by light, or is there a light-independent component? What is the minimum intensity of nighttime light needed to shift sleep timing in people with different levels of artificial light exposure? How do individual differences in circadian type, genetics, age, and health status affect sensitivity to lunar light? And what are the long-term health implications, if any, of living in environments where the natural monthly light cycle is effectively erased by artificial lighting? These questions are not unanswerable, but answering them requires research designs that have not yet been widely implemented. Large scale studies, so tracking multiple physiological parameters continuously across many months in populations with varying light environments, combined with genetic analysis and careful statistical methods, would go a long way toward resolving the current uncertainty. The technology to conduct such studies largely exists. What has been lacking is the combination of funding, scientific coordination, and methodological consensus needed to make it happen at the scale required. What the existing research does establish clearly is that the full moon is a real physical event with real measurable effects on the light environment, the gravitational environment, and the behavior of a very wide range of living systems. It establishes that human biology contains timing systems that are sensitive to light and that evolved in an environment where the monthly lunar light cycle was a constant presence. It establishes that some studies have found small but consistent associations between lunar phase and human sleep, and that these associations are larger in populations less exposed to artificial light. And it establishes that the mechanisms proposed to explain these associations, including direct light effects on melatonin and circadian timing, are biologically coherent and consistent with what is known about human physiology. What it does not establish is a clear, replicated, mechanistic account of how the full moon affects the human brain every month. The gaps in that account are not evidence that no effect exists. They reflect the genuine difficulty of studying small, cyclically varying biological signals in organisms that live in complex, variable, and increasingly artificially lit environments. The moon's relationship with human biology sits at the intersection of physics, neuroscience, evolutionary biology, and ecology. None of these fields alone has the tools to resolve the question, but together, with carefully designed research, they're moving toward a clearer picture of what the full moon actually does and does not do to the biology of the creatures living beneath it.